Hello and welcome to the Thesis Writer's Guide to Making Professional Scientific Figures. This is part 2 of a two-part series. This part focuses on image editing for students working heavily with scientific images. If you have not yet watched part 1, please do. It contains basic and essential information that I'm not going to repeat here. So let's begin. For those of you working with images, you probably are already familiar with some of the common file extensions. We're going to start off by talking about TIFF and PNG file extensions. Those two file types represent lossless forms of image compression. What that means is that whenever the computer saves your image file, it looks for shortcuts and ways to reduce the content of the file itself, but never at the cost of sacrificing the image quality. So what we recommend is that you use TIFFs when you acquire the image and keep that as your original master file. That way you know that the quality is always going to be the same and in its highest level. Whenever you make an edit or you manipulate your image, it's good to save that edited version as a new copy, and you can save it either as a TIFF or a PNG. That's going to preserve the quality. If you make an edit and you don't like how it affected the image, you don't want it to affect the original. Sometimes these edits are permanent, and that's why we always make a copy when we do our manipulations. The other category of file extensions are those similar to JPEG, and those are the lossy compressions. And these are compressions that are more aggressive, and when they do their compression, they are actually reducing the quality of the image. And sometimes it's worth it because you can get a very small reduction in the image quality, but a very large reduction in the file size, something like tenfold. And in fact, we are going to use this format whenever we convert our figures or our Word document into a PDF. It will automatically convert the images into JPEG. But we're going to save this conversion to JPEG for the final step so that it only happens once. We do use JPEGs whenever we're showing our figures either on the web or in PDF format, but as long as this is just one final step at the end, it's going to have a negligible effect on the image quality. So just as an example, here's a picture of a neuromuscular junction as a PNG. It's 178 kbs. Here's another one saved as a high-quality JPEG. It's 11 KB, so a tenfold reduction in size, and it's very difficult to see any differences between these images. And I should say that the JPEG has a range of different compressions. Some of them are high quality and some of them are low quality. So we have to be a little careful when we're saving JPEGs that we always choose the high quality compressions. This is a low quality compression. The file size is reduced another twofold. It's only 5 KB now, but you can really see the compression artifacts very clearly in the image. We don't want that to happen. In terms of what we're going to do with our images, one of the things you definitely want to look into is editing them to kind of increase the visual effect that the image has for the viewer. So the viewer is going to be looking at it with their eyes, and you want to make the image pop out, stand out as much as possible for them. And the way that we do this, the way we can start thinking about it, is to remember that these images are made up of a block of square pixels, and each pixel is just a grayscale intensity value. In this case, our images, if we zoom up on a set of these pixels, have a value could potentially have a value between 0 and 255. That's the range that an 8-bit image holds. 8 bits, a bit is just a 0 or 1, so there's two different states it can be in. 2 to the 8th power is 255, so by definition an 8-bit image has 255 different intensity values that the pixel can have. You could save your image as a 12-bit or a 16-bit image, and oftentimes in science we do that. If you save it as a 12-bit image, it's going to have 2 to the 12th intensity values. That's about 4,000. Or 2 to the 16th for a 16-bit image, which is about 64,000 different intensity values. Now that is typically overkill. The human eye can't really distinguish a 12-bit or a 16-bit image. We're kind of limited to about 255. So for most applications, an 8-bit image is fine when you're acquiring the image. But if your lab does a lot of image quantification or analysis processing, you might consider using a higher bit depth image. But for this presentation, we're just going to assume everybody's working with 8-bit images. And what I want to show you here and point out is that the pixels with a value of 0, those are pure black. There was no photons that got captured by the camera in that spatial area, whereas the brighter ones have a higher intensity value. In this case, the highest value only goes up to 121. because the pixels in this set that I chose don't represent the brightest pixels in the object itself. And that's often the case. So we can take a look at our histogram of pixel values, so we can graph all the pixel values and show them as a histogram using lots of imaging editing software. We're going to show you that in a second. But this is what that graph would basically look like. You have your pixel intensity values from 0 to 255 for an 8-bit image. And then we have the number of pixels within our image that have that particular value. So, the most common value here would be something around 100. The maximum value I showed you before was about 120, and there's some pixels that go all the way down and have a 0 value. Now, if our histogram looks like this, what we can do to help the reader is adjust the pixel values so the viewer can see more of the information that's in there. And we do that by stretching the pixel values to cover the entire display range. So we take a histogram that looks like this and turn it into a histogram that looks like this, where our highest pixel value is set to 255, our lowest one is set to 0, and our other values are remapped linearly along the 255 different intensity value possibilities. Now, I should say that the most ideal thing to do is to be monitoring the histogram while you're taking the image and adjusting the settings of your cameras or your microscope so that you fill up this histogram completely. That way you ensure you get the most information from the object that you're imaging. But sometimes that's not possible for technical reasons. Maybe our sample isn't bright enough to get all the way up to 255 value, or we've cropped an image that was larger. It did contain higher intensity values, but now our cropped version doesn't have those values anymore. That was the case that I showed you in this example. So when you find yourself in that situation, you can do some post-processing to really increase the brightness of your pixel levels. What the ultimate effect is, this is the original set of pixels, and this is the effect after we've remapped them to cover the whole display range. And hopefully what you can appreciate here is that now the brightest pixels are set as pure white, the darkest ones are still pure black, and it's a little bit easier for the eye to distinguish the differences and intensities of these pixels that are in the mid-ranges. So that's the goal of these adjustments. And we can do these adjustments either in Photoshop or GIMP. Photoshop, you may or may not have access to. It can be expensive. But GIMP, on the other hand, is freeware, so you can download that for free. It's available to anybody, and it has the same capacities for the sake of this talk that Photoshop does, so it can do everything that we need to do to get our images to look good. So if you don't have access to Photoshop, feel free to download GIMP. In either case, you're going to have an option to look at your pixel histogram of a given image, and what you're going to see in Photoshop and in GIMP is your histogram and 3 different triangles underneath it. And these are the triangles that we're going to use to stretch our pixels over the display range. We've got one triangle here on the left that's going to set our 0 value. So we're going to move that to the right to set our pixels to 0. And we've got another triangle here all the way on the right. We're going to move that to left to set our highest pixel values in the histogram. And effectively what that looks like here is you're going to move that left triangle, the 0 value, to the right. And you're going to turn all those pixels to the left of that triangle to 0. And that's okay as long as there's no pixels that had any of those values in your original histogram. Otherwise, what you're going to do is you're going to take those values and essentially reset them to 0, which is not what you'd want to do. But if your histogram doesn't start until the values get higher, then you can slide that triangle forward and just get rid of that excess wasted space on the left side of your histogram. And the same idea for those pixels on the right. You'll move this triangle to the left to set your first pixel at that right edge of the distribution to 255. And then you're going to remap all those other pixels linearly in between those 2 new endpoints. And some tips here are, when you're moving that left triangle right, leave some background, some non-black, to show the relative brightness of your object of interest compared to everything that's around it. I'll show you that in a second. Don't saturate. Tip 2 is don't saturate the pixels and objects that you care about. Otherwise, that information will be lost. And by saturate, I mean moving either of these triangles across pixel values of pixels that are in objects that you care about. So you're going to lose any of the information that's actually contained here. You're going to set all of these to 255. It's going to look like there's no difference in those pixels, even though there was actually differences in the pixels there. So we'll be a little bit careful about that. And then the last tip is for multicolor images, work on each color channel separately. So our multicolor images are going to be red, green, and blue. We're going to have 3 different channels, and we can edit those channels separately makes it easier to see what you're doing when you're doing this type of work. Before we get into a demonstration, I just want to mention that when you do image quantification, you want to be sure that you're quantifying the original image, not the manipulated one, and not the manipulations that we're doing here. This is just for display purposes. We're just making it easier for the viewer, the reader, to see the information that's contained in the image. But it is changing the pixel values in your image, and that would have a bad effect if you were trying to quantify images, because you're no longer working with the original. So make sure you have an original copy and that you use that one for any quantification that you're going to do. The last triangle I want to talk about before the demonstration is this middle one called the gamma. And we can move that triangle around to bring out the midtones in our image. And what this does, as we shift it around, is it's going to create a non-linearity in the mapping of our original pixels with our new display pixels. And that is useful because if we have a pixel value, say, of 120, and we remap it by moving the gamma around, we can see that we can really boost the pixel level of this midtones without having much effect on those pixels that are at the ends. So we're not going to saturate or drastically alter those pixels at the 0 or at the 255, the extreme ends. We're only going to bring up those midtones. And we'll see that that ends up being really useful to help make our images look pretty bright and vibrant. So I'm just going to show you how easy this is. If we go to Photoshop here, I can open an image that I have on hand to work with, and you might think this image already looks pretty colorful and pretty decent, but there's still a huge amount of improvement that we can make. I'm going to duplicate it so that we can see the before and after, and let's work on this one. Now, I'm going to go to Adjustments, Levels. That's how we get to the histogram. Because this is a multicolor image, it's got a red, green, and a blue channel, so we're going to work one by one. Here we've got the red, and when I move this 0 triangle up to kind of the peak, the first peak of this histogram here, you can see that the background is getting reduced. As I said, you want to leave a little bit of background in there, so don't go crazy and kind of get rid of it. People want to see the contrast between your object and the background, so you've got to be a little light-handed when you're doing this. You just maybe move it a little to get rid of anything that's unnecessary. And then we can do the triangle to the right here to set our brightest pixel values. As we can see, these pixel values are already pretty bright. It looks like there's even some saturation here in the original image, so I'm just going to leave them the way they are. But we can adjust the gamma to bring out the midtones a little bit and make that a little bit brighter. Let's do the same thing with the green. The green doesn't really have any background. It's already, we're getting a lot of pixels there at the 0 value. But we can bring up make some of the higher pixels more intense by moving this triangle forward till we get to about the start of the ramp up of significant counts in our histogram. And we can also play around with the gamma a little bit to bring out some of those midtones. And here we're just eyeballing it to see what looks good, what's helping us distinguish different colors in the images. And I like it about there. And then we do the same thing for the blue. Again, there's not much background in the blue channel, but we've got most of our bright blue pixels. We don't really start setting them at a significant number of them at a high value until we're down to about 147 of an intensity value. We can set it there, and then we can use the gamma to bring up those midtones a little bit. Whenever you're happy with it, you click OK. This is a permanent alteration of the image, so if we go back and look at the histogram after I click OK, you can see that now the histograms get redrawn according to these new endpoints and gamma values. So be sure you have your original image unmodified so that if you don't like it or it becomes problematic, you can always switch back to the original. And now just to compare our effect, we can look at the first version and the second version, and you can see that that has had a big impact. I think especially bringing out the blues and brightening the greens a little bit makes this image a lot more vibrant and helps the viewer distinguish some of the color identities a little better that we have in there. Okay, so that was quick. It only takes 10 seconds, and you can really do make some nice improvements to how your images look. Now that's one of the big things we're going to do with images. The second big thing we have to be aware of when we're working with scientific images is how do we know that we have sufficient resolution to print our images out and have them look good in the printed copy. And as I said in the last part in this series, a normal printer needs about, at a minimum, 220 pixels per inch. But typically our slideshows through our projectors and monitors are only showing us 72 pixels per inch. So we don't really know how it's going to look when we print it out until we print it out. But there's some really easy steps you can do to set the resolution of your images so you know exactly what they are. And I'm going to show you those 3 steps now. Step 1 is simply to open the image in Photoshop or GIMP and manually set the resolution to 220 pixels per inch and determine the true size of your image. So let's just look at a toy example here. If we have a block of pixels and there's 100 pixels there, 10 rows and 10 columns, and our resolution is set at 10 pixels per inch, then this image is going to be 1 inch by 1 inch. So we have 10 pixels by 10 pixels at 10 pixels per inch. If we want to set our resolution to a higher resolution, like 100 pixels per inch, what's going to happen? What's going to happen to the image size? Is it going to get bigger? Is it going to get smaller? Well, we're going to have the same number of pixels. We're not going to create or destroy any information, but we're going to make the density of those pixels higher. So what's going to happen is the image is going to be reduced in size. So now we've taken our 10 pixels by 10 pixels and put it into a smaller area. So now it's 1/10 of an inch, and that gives us 100 pixels per inch. But in order to get that resolution, we've seen that our image size is now much smaller. So the price you pay is the size of your image to get that higher resolution. When we change our pixels per inch, we're changing the image dimensions. We are not changing the pixel number. That's a very important point. Let's take a look at how you do that. Again, I'm just going to use Photoshop. Let's open a version of this that's 72 pixels per inch. When we go to image size, and I should mention that I have a PDF posted on the web so you can get these instructions of where to find these commands very easily, either in Photoshop or GIMP. But when we go to image size, we get all the information about our image. We see the size of the image, we see the total pixel count, we see the image dimensions, about 6 inches by 7 inches, and we see the resolution, 72 pixels per inch. So, finally we can see the resolution of our images. What we want to do is set this at 220 and see what the size of the image really is. And an important thing to do here is to uncheck the resample box. Resample box is going to try to add pixels or remove pixels, and that is not what we want to do. We simply want to change the resolution and see what the true dimensions of our image are at that resolution. And if I set this to 220, then I see my pixel, same pixel numbers, is now 2 inches by 2 inches instead of about 6 by 7. So that is the size of the image that I have to work with if I want to use the resolution of the image that I need to print out at 220 pixels per inch. So what I would do is I would just save this image and insert it into my PowerPoint, and it will open at its new image size and with the image resolution that I set. I have already saved it, so just to show you that, I'm going to insert those 2 images here. So here is the 72 pixel per inch version, that's 6 inches by 7 inches. And now let's insert the 220 pixel per inch version, that's 2 inches by 2 inches. So PowerPoint, whenever you go to insert picture, is going to preserve the resolution of the image, and it's going to appear at the image size that we want. And if we know what we set it at, then we know the resolution of our image, and we know what it's going to be, and if we can use it. So now something comes up where we have to think, okay, so now I know the dimensions of my image, but is it the right size for my figure? Remember that your figure, if you set this up the way that I told you in the last part, is going to be about 9 inches by 6.5 inches, so you get a sense of scale for how big you want your figures to be. And if 2 inches by 2 inches is big enough for what you want to show, then you're in luck. So you basically insert your 220 pixel per inch image into PowerPoint. The image will appear. If the image is bigger than you need, then I'm going to show you how to handle that. That's an easy problem to solve. If the image is smaller than you need, then you've got some thinking to do about whether this is the right image to show or not. So let's first talk about what happens when the image is bigger than what you need. In that case, all we have to do is decrease the image dimensions and reduce the pixels after we decrease the dimensions. So again, let's take our toy example where we have a 10 by 10 block of pixels at 10 pixels per inch, which means the image dimensions are 1 inch by 1 inch. But let's say we only need our image to be a ½ inch by ½ inch. We're going to keep the same resolution. We're just going to make the image dimensions smaller. What we can do in PowerPoint is use the bounding box, use the handles to drag it to make it smaller, right? And what that would look like is basically we could take this block of pixels. Let's just copy it. Get rid of some of these elements that we don't need. All right, so here's our block of pixels, and we're going to take this and we're going to make it half the size. When we do that, what we see just by dragging these handles is that it's going to take those pixels and increase the pixel density. So it's not going to get rid of the pixels. They're still going to be there. It's increased our resolution of our image. So now instead of having 10 pixels per inch, we actually have 20 pixels per inch because we have 10 pixels in ½ an inch. And that's fine. That's extra resolution. We don't need all that. The only downside to that is it can end up making your files bigger than they need to be. So if you find yourself in a situation where you have file sizes that are very large, more than 5 megabytes per individual slide, then what you might think about doing is going to use the format compress picture to reduce the pixel size back down to the resolution that you want. In our case, in the real life, it would be 220. In this example, it would be 10 pixels per inch. So we'd throw away those extra pixels to get it back down to the resolution that we want. And you can see that's pretty easy to do. So if I insert an image in here, and I end up needing a smaller version of it, so I shrink it down by dragging it with the handles, then I can just go to File, Compress Picture, choose what resolution I want, and tell it to just do it for this selected picture, click OK, and it's going to throw away all those extra pixels and make my file size smaller. So that's always an option if you're worried about file size. So that's the easy problem whenever your image is bigger than you need it to be. The harder problem is when your image is actually smaller than you need it to be, right? So in that case, what we'd want to do is increase the image dimensions, but that means to keep the resolution the same, we'd have to add pixels. So in our example here, let's say we've got our 1 inch by 1 inch, 10 pixels per inch image, and we want to make it 2 inches by 2 inches and keep 10 pixels per inch. We can't do that because when we make this bigger, if we tried to drag it, it would reduce the resolution of the image. It would go down to, in this example, 5 pixels per inch. If we tried to make it 10 pixels per inch, we need to add in pixels somehow, right? And those pixels weren't in the original image. So they have to be created, they have to be made by the computer itself. And PowerPoint will do this, so you need to be careful. If you're trying to add pixels to give your image better clarity or resolution like they do in CSI Miami when they zoom in, it's not going to work. That's not how images work. So you either need to recapture the image at higher resolution, or you can make the image bigger, but you have to make sure that you preserve the pixel boundaries so that you're not misrepresenting the information that's in your image. And this can be useful. Sometimes you do want to zoom in and show the details that are there, but you don't want to do it by dragging, because what PowerPoint is going to do is it's going to add pixels in a way that makes it difficult to see where the original pixel boundaries were. So if we take a look at that, when we're enlarging our microscope pictures, don't add fake pixel information. It's called interpolation into your image. When you're enlarging, you want to show the real data. So if you were to drag the bounding box by the handles to resize, you are going to interpolate your image, and that's not what you want. Let's say, for example, you wanted to show this tiny piece of an image, which is only about a 1/2 inch by a 1/2 inch at this resolution, but you wanted to make it, I don't know, 2 inches or 3 inches by 3 inches. You can do that, and you can do it in a way where you preserve those original pixel boundaries, and you can see that it's looking good, but it takes a little extra effort to do that. If you were just to drag it in PowerPoint, you would see that it would add in pixels to try and create information, and it ends up looking very blurred. And scientists can spot these kind of interpolated images from a mile away. You get used to how they look, and it's quite offensive to their sensibilities because now they don't know what the limitations are of the information content of the image. So don't do that. Don't drag it to make it bigger. Instead, we're going to use no interpolation or a nearest neighbor algorithm in Photoshop or GIMP to add in pixels that preserve the boundaries of those original pixel lines. And I'll show you this is also pretty straightforward. So let's open that original image in our image editing program. Let's crop a tiny piece of it that we're interested in. So this little protrusion off of this synapse. And we're going to crop it, and we'll zoom in. And when we zoom in Photoshop, it's preserving the original pixel information that's there. And now all we have to do is go into our image size mode, and we want to add in more pixels, but we want to do it by this nearest neighbor method to preserve the hard edges of the original pixel boundaries. And we can make it, however many pixels we want, and we can change the size as well. So we can make it 6 inches by 3 inches at the resolution that we want. So we had to add a lot of pixels in order to do that. But we added the pixels in a way that's going to preserve the original content, information content of the image. So when we zoom out, all we really did was add more pixels to this, but those pixels are going to preserve those pixel boundaries, the original ones. So we're showing the content that is there in its original limited form, but we're making it easier for the people to see the fine details of the image. So then again, you would just save that and insert it into your PowerPoint. Okay, so that is the final thing that I wanted to show you, and with that, I think we're done. So good luck. Like I said, there's specific instructions about using PowerPoint and GIMP on the website, so if you do want to do any of these editing steps, please check them out. And as I said before, if you have any questions, feel free to contact me. My name is Ryan Draft. I'm the academic advisor in the neurobiology department. So good luck and have fun.